All right, sorry I didn't make this video earlier. I probably should have. I realized there is some confusion, so let me try to help. The nickel lab is a lab that helps you relate energy and light to a property of a solution. You're going to have a solution that has nickel in it. That solution has a bright greenish color, and you're going to determine how much nickel is in an unknown sample by using what's called a calibration curve. A calibration curve helps you calibrate how an instrument responds to a particular analyte, in this case, nickel 2. I just wanted to start by a little brief information about why do we see color. Here's our color bit wheel, Roy, G, Biv. So if you look at a sample and it appears red, really what's happening is that the sample itself is absorbing green light. You see color because chemical species absorb certain wavelengths of light. We're actually really starting to get into this in our new unit. We started with light, so I did put a little bit of this in your lecture notes already. The light that's reflected, that is to say not absorbed, is the color that you see. The lecture notes have a fun example about why are fall leaves orange and green, or sorry, orange and yellow, and summer leaves are green. So relating to chlorophyll and other compounds in the leaf and how they absorb and reflect different wavelengths of light. So why does your nickel 2 solution appear to be green? So what you see is green. You're seeing green, so that sample is absorbing, nickel 2 solutions are absorbing red-orange light. We're going to use that absorbance property to determine some things about your unknown nickel solution. The first type of calculation you were asked to do in this lab was a dilution calculation. I think this is now 5.4 and this page isn't relevant, so I'm sorry, that's an old reference to the older textbook. But remember, there's a shortcut for dilutions. You can take the molarity of the first solution and the volume that you use of the first solution to determine the molarity of the second solution if you know how much of the second solution you're making. In this case, you're making a new solution from a stock solution that is pretty concentrated. So this question is asking you, how would you prepare 25 mils of a solution that's 0.140 molar from a stock solution that's 2 molar? So in this case, we know the second molarity. We know we want a 0.140 molar solution. We know how much of it we want. We know we want 25 milliliters. And we know the molarity of the original solution. So we can figure out how much of that original solution we want to use. So V1 equals M2 V2 over M1. So we can plug in our values from the problem. 0.140 molar times 25 milliliters divided by 2.00 molar. So molarities are going to cancel. And when I solve, I get 1.75 milliliters of solution 1. And we're going to dilute that amount to a volume of 25 milliliters. That's how we would prepare a dilution. So you're having dilutions in this lab. You're going to make a series of dilutions so that you can build a calibration curve to determine the response of our spectrometer to that nickel-2 analyte. We're going to be using Beer's law in this lab. Beer's law helps us relate the absorbance of light to the concentration of the thing that's absorbing light in solution. It takes the form A is absorbance equals epsilon. This is a molar absorptivity, big fancy word for how does this substance absorb light. Molar absorptivity constant. I will say it is constant for a particular analyte. It is different for different analytes. So in this lab, you're going to determine the molar absorptivity constant for nickel at the wavelength that you're measuring. So it's constant for an analyte at a wavelength. So you're going to actually measure 3 different wavelengths, and you're going to calculate 3 different molar absorptivity constants for this lab. Okay, A is absorbance, epsilon is that molar absorptivity constant. B stands for the path length of the cuvette. That's the little cell where you put your sample. And concentration of your analyte is C, so concentration. In this case, you're going to be finding the concentration of nickel-2 that's in solution. So we're going to be relating to amounts of nickel-2 in solution. So absorbance changes with wavelength. You're going to be measuring at 3 different wavelengths. Your concentration that you calculate should be the same for all 3 wavelengths, or roughly the same. Your molar absorptivity constant might have some variation, because here's our absorbance spectrum for nickel-2. You can see that this compound doesn't absorb all light equally. It absorbs some light better than others. So here is what's called a maximum at 395 nanometers. That's the wavelength of light where most of the solution is absorbing. There are 2 other maxima, so 658 nanometers and 722 nanometers. All of these are good wavelengths to measure this solution. These are all called maxima, but you're going to absorb more light the larger the maximum is. So 395 is going to have the most absorption. These other 2 are good absorption wavelengths, but they're not going to absorb as much. Your molar absorptivity constant is going to be different for all 3 of those wavelengths because the substance absorbs light differently at all of those wavelengths. Okay, I think that to be true. Actually, I've never run this lab where you're calculating the molar absorptivity constant. I'm not sure how much it varies between wavelengths, but we'll see with your experimental data now, won't we? Okay, let's talk about the calculations for the lab. So absorbance maxima. So the maxima in this plot are the best wavelengths to choose because that substance is going to absorb well in those regions. Your concentration of nickel is the same in all the wavelengths, but your absorbance value is going to be different, which is why we need 3 different calibration curves. So the unknown will have a particular concentration. But that concentration will have a different absorbance depending on the wavelength of light. And in even saying that sentence out loud, I just answered my own question. So here's Beer's law: absorbance equals epsilon, beta, and concentration. If we know our unknown has a particular concentration, but it's going to absorb light differently, if absorbance is going to be a variation, then absorptivity must change with the wavelength, because the path length is going to be constant and the concentration is going to be constant. So if absorbance changes, then it must be epsilon changing. So that epsilon is unique for a given wavelength of light that that substance is absorbing. Okay, you're going to measure absorbances of all the standard solutions to make curves for each different wavelength, and then you're going to determine the concentration of your unknown for 3 different wavelengths. So you're going to prepare data tables in your notebook. I'm going to encourage you to look at the worksheet linked on Canvas for some ideas about the data tables that you'd want to put in your notebook. But you're going to record data for standard solutions, solutions that we know the concentrations of, at 3 different wavelengths. And you're going to use those to build a calibration curve. You're also going to record your unknown concentration, or sorry, your unknown absorbance at each wavelength. And so not only are you going to record your absorbance data for your standard solutions, you should also record your volumes and concentrations for your standard solutions. Your pre-lab worksheet is pretty detailed in what you need to put in your notebook. So I will just highlight that again and say, look at that worksheet that's in the lab report. It'll tell you what data you need to put in. Where students seem to be getting confused is this idea of calibration curve. They don't feel that that is something that was really discussed that well. So let me talk to you about a calibration curve. Again, a calibration curve is a curve used to set an instrument's response to an analyte. So basically you're building this curve to determine how much or little your instrument responds to a particular analyte. In this case, absorption of nickel 2+ ion in aqueous solutions. So we're going to calibrate our instrument's response to nickel 2+. Our calibration curve is going to have X values and Y values. On the X axis, you're going to plot concentration. And that concentration is going to be molar concentration, big M. So what is the molarity of nickel 2+? The Y-axis is going to have your absorbance values. You're going to plot the absorbance at different concentrations. If you were to plot a point that, say, is clearly off of this line, that would be an outlier point that we would exclude from our data set. In this particular experiment, we took all the data for you, so we know that the measurement should give you a linear relationship, because if you notice it, Beer's law is actually going to be an equation that has a linear relationship. Let me show you how. So let's think about the equation of a line, y equals mx plus b. In this case, your y values are absorbance. So when you plot the Y value as absorbance, your X value is concentration. Your M, the slope of that line, is actually epsilon times beta. So your epsilon and your beta should be, or sorry, not beta, epsilon B. Your epsilon, your molar absorptivity constant, your path length, those should stay constant for a given wavelength. So let's say this particular one is our line for 722 nanometers. You're going to have 3 different lines on your graph. You're going to plot 3 different lines on the same graph. So let's say this one is the 600 and something. What was that wavelength? 658 and 395. And let's do a third color. This is that 395 nanometers. So you're going to put 3 different lines on the graph. They should all be trending towards, and I didn't exactly draw this correctly, they should all be trending towards the origin. At 0 concentration, you should have 0 absorbance. So forgive my artistic rendering here. I was not perfect. The other thing you're going to be plotting on your graph is a graph at absorbance equal to 0.1 and a horizontal line at absorbance equal to 1.0. These are some boundaries. We do not want you to plot data that is below 0.1 absorbance or above 1.0 absorbance. Beer's law only works between it only has that linearity relationship when absorbance is between 1 and 0.1. Below 0.1 or above 1 are out of the linear range. So when you're making your plot for this lab, I want you to delete any data points where the absorbance is below 1, or sorry, above 1 or below 0.1. Okay, now, having said that, we are going to generate 3 trend lines. We're going to generate a trend line for each wavelength. You're going to show the equation on your graph. The Excel how-to videos should show you how to do those. Let me show you what you're going to do with the trend line. So again, y equals mx plus b. That's definitely true. So your trend lines that you generate for this experience are going to be in the format of y equals mx plus b. Well, you want to calculate the molarity of your unknown solution. That's the concentration. That's the variable x. That's what you're going to calculate for. So you're going to plug in the absorbance at a wavelength and solve for x. So you should get 3 different concentration values, 1 for each wavelength of measurement, but they should be roughly the same. If they're not, you want to check your math because that nickel concentration is constant for all 3 wavelengths. It's just absorbing a little differently because those maxima have different heights. Okay, so you should solve for 3 concentrations of nickel at each wavelength. They should be roughly the same. How are you going to then calculate molar absorptivity? Well, here's Beer's law again. You want to know the molar absorptivity. The experimental data should have given you the path length, the concentration that you just solved for, and you have the absorbance for that trial. So you should be able to solve for the molar absorptivity based on the absorbance and the known concentration and the path length. Okay, in Excel, this is sort of a checklist. Make sure that you plot X is going to be concentration of nickel for your calibration curve. Y is going to be the absorbance values. So make an X series, make a Y series where X is nickel concentration, Y is absorbance. You're going to adjust that series to remove any dots that are above A equals 1 or below A equals 0.1 because those are outside of the linear range. You're going to label the series. So label them with wavelength 395 or wavelength 658 or wavelength 722, something that tells me which line corresponds to which wavelength. And when you're solving up here, I'm going to put this in another color, make sure you plug in the absorbance for the correct wavelength. That's the most common student mistake, is that students they plug in the absorbance maybe at 395, but they plug it into the equation for the 722 line. So make sure you know which line corresponds to which wavelength and that you're plugging in the absorbance that was measured at that wavelength. You want to add the lines for the actual value. So add those horizontal lines at 1 and 0.1. The videos will tell you how to build a horizontal line. You want to add trend lines for all 3 wavelengths. You want to know which trend line goes with which data series. So be very clear about that when you build your chart. You're going to label the axes and make a title. The videos will show you how to do both of those things. Remember, the x-axis is the concentration of nickel in molarity, and the y-axis is absorbance, which has absorbance units. And lastly, you're going to adjust the minima and the maxima of the X range to make sure you've maximized the plot. I don't want a bunch of white space. I want to mostly see your data in the plot. Do let me know if you have any questions about this use of Excel, because this is kind of a new use of Excel for us. All right, I hope this is helpful. Thanks for listening.